So I'm really happy to welcome our first speaker, Dr. Sarah Whittle. She's a professor at the University of Melbourne, and she will be covering the developmental stage of the lifespan. The floor is yours.
Good morning, everyone. Thank you for showing up nice and early on a Friday morning for this symposium. So I'm going to be presenting a bit of an overview of some work that I've been involved in looking at pubertal influences on brain development, how they interact with the social environment, and how all of these things kind of interact together to influence sex differences in behavior and mental health. So we first learned or kind of clued into the idea that maybe there are sex differences in adolescent brain development back when some of the first MRI studies of adolescents came out in the 90s. So you can see from this figure here, this is showing trajectories of grey matter and white matter across adolescents. The arrows there point to the peaks of grey matter volume in females, the red lines, blue, are the males. So you can see there that we saw that there were earlier peaks in brain development in females relative to males. From this point, it was speculated that perhaps this had something to do with puberty, because these sex differences in trajectories nicely mirror what we know happens in puberty, with females typically developing a couple of years earlier than males. These sex differences in brain development trajectories have been quite a consistent finding over the years. This is just highlighting a couple of figures from a review paper from a few years back. The top there is showing sex differences in structural brain development. The bottom sex difference is in functional brain development. So like I said, from this early work, we thought that maybe puberty had something to do with shaping sex differences in brain development. We're still a little unclear on exactly why that is, but we think it's probably something to do with hormones, or a combination of hormones and psychosocial factors. But we know that all of these things seem to shape brain development in sex-specific ways. So there's now quite a bit of work out there that has looked at associations between pubertal development and brain development. But most of this work has focused on the physical signs of puberty, and most of the work has looked at group-level associations, so what happens kind of for the average adolescent. And there's less work that's looked at individual differences in pubertal maturation. So I'm going to focus on that quite a bit today. So in particular, looking at things like pubertal timing. So this refers to someone's pubertal stage relative to their same sex, same age peers. And pubertal tempo, which refers to individual differences in the speed of pubertal development. So there's emerging work now that indicates that these individual differences in pubertal maturation are important for sex differences in brain development. And there's also emerging work to show that these individual differences in pubertal maturation likely interact with other factors, for example, things in the social environment, to predict brain development. And that's what I'm going to be focusing on. So in terms of mental health, which is what I tend to look at in my work, I'm interested in how brain development confers risk or resilience for mental health problems. But in terms of individual differences in pubertal development, it's a fairly consistent finding out there now that early pubertal timing seems to confer risk or vulnerability to mental health problems, particularly in females. The findings are a little more mixed for males, with some studies showing earlier timing and other studies showing later timing might put males at risk. A recent systematic review that I did with a colleague, we showed here that particularly girls with pubertal timing seem to be more vulnerable to mental health problems as a result of increased experience or sensitivity to negative social environments. So we saw that there were kind of complex interactions between pubertal maturation and the social environment, and in particular, negative or aversive environmental exposures that predicted mental health in females. So like I said, I'm going to kind of give an overview of some of the work that we've done looking at the role of the brain in these associations. And all of this work has used data from the ABCD study, the Adolescent Brain Cognitive Development Study, which I'm sure probably most of you are familiar with. For those that aren't, it's a really large-scale adolescent development study that's happening in the US. It enrolled over 11,000 early adolescents, aged 9 to 11. Those adolescents are being tracked with MRI every 2 years, MRI of the brain, and other assessments happening either every 6 months or every year. On the right there, I'm just showing you here the pubertal developmental trajectories so far in terms of what data has been released from the study. So females are in the top graph there, males on the bottom. So this is a fairly typical pattern of pubertal development across adolescents. And the dot there reflects the midpoint of pubertal development. So you can see that for females, their midpoint of puberty is earlier than males. It happens at about age 12. Whereas for males, it's between age 13 and 14. So the first study I wanted to talk about is some work that's being led by Divyangana Rakesh and her colleagues at King's College London. So here we were interested in looking at how aversive environments impacted individual differences in pubertal maturation, which in turn impacts brain development and also mental health. So here we were focusing on a measure of income to needs, so I'll refer to it as poverty from this point forward. So we found that poverty was related to greater cortical thinning across adolescents, and that's a fairly well-replicated finding. Kind of more aversive environments tend to be associated with kind of accelerated brain development, so that's what we saw here. But when we looked at whether individual differences in pubertal maturation mediated those associations, we found some interesting things. So we found that poverty was related to early pubertal timing, which is also a fairly well-replicated finding. And then early pubertal timing was in turn related to greater cortical thinning. So kind of faster earlier pubertal maturation, faster brain development. Looking at the pathway through pubertal tempo, though, we found that poverty was related to slower pubertal development, so a slower tempo, and that slower tempo was related to slower brain development, so reduced thinning, less thinning. Some really interesting, complicated kind of opposite patterns. So we looked at the figure at the top there is for mean thickness, so whole brain thickness. We did look regionally as well. So the plot there in B, or the figures in B, show the effects of timing. C show the effects of pubertal tempo. So there were some regional effects there too. And then interestingly, when we looked at the full pathway from poverty to pubertal maturation to brain development to mental health, we found a significant indirect pathway for internalizing symptoms. And again, this was quite interesting. So the indirect pathway was through pubertal timing, not tempo. So the finding was that greater poverty, earlier pubertal timing, greater thinning, which is what I showed you on the last slide. Interestingly, greater thinning was related to lower internalizing symptoms. So there was actually no total effect of poverty on mental health, but there was this indirect pathway that kind of reflected almost an adaptive mechanism following poverty. These significant effects were only found in females. There were a couple of the individual paths that were significant in the males, but this full mediation model was only significant in females with no indirect effects found for males. I'm going to switch over now to another study. So in this study, we were interested in stressful things that were happening to the adolescent during their development. And here we wanted to kind of look into this notion of sensitive periods. It's been suggested that puberty in general might be a sensitive period of brain development, high neuroplasticity, potentially increased sensitivity to the effects of aversive environments on the brain. But puberty, of course, is not a unitary construct. It's comprised of different stages of development. And there's been a little bit of work suggesting that maybe certain pubertal stages of development are more or less sensitive, engender more or less sensitivity to the environment. So this is what we looked at here. We looked at the interaction between pubertal development, so Tanner stage, and stressful life events in a longitudinal manner, looking at effects on brain function here. So here we're looking at activity during emotional NBAC task. So this is quite early days for these findings. We haven't really kind of wrapped our head around what they actually mean. So if anyone has any ideas, I'd love to hear from you. I'm not showing you all the results here. I really just wanted to highlight that we found very different patterns of associations for males and females. I'm just showing a few of the significant ones here. And like I said, we haven't fully interpreted what these mean, but you can see here, so the different colors here, the different trajectories represent different Tanner stages. So the pink and purple lines represent later pubertal development, so stages 4 and 5. The blue, green, and orange lines represent earlier development, Tanner stages 1, 2, and 3. But you can see there for females, for example, for the middle frontal gyrus, those in earlier stages of puberty seem to be more sensitive to stress or adversity they're experiencing. That slope is significant for those earlier pubertal stages. Whereas males, for example, on the other side, those in the later stages, so Tanner stages 4 and 5, were more sensitive to stress. So those slopes were significant whereas the early stages were not. So some really interesting and kind of complicated patterns of sex differences. I'm going to switch over now to talking about hormones and just talking to you about some of the work we're doing now looking at hormones in the brain in the ABCD sample. So this work I'm presenting has been done by my student Muskan Ketan, who's here at the conference, and she presented some of this work in a poster the last couple of days. You might have seen it. So in her first work, we wanted to take a look just cross-sectionally, what are the links between estradiol and brain structure and function? Out of all of the pubertal hormones that have been looked at in relation to brain development, estradiol is one of the kind of the hormones that has been looked into the least. So we wanted to take a look at this in females specifically. Estradiol samples are not available for males in the ABCD sample, so that's a limitation that we can't look across both males and females. But here we were interested in the female brain in any case. So in this work, Muskan applied elastic net regression to investigate links between hormone levels and brain structure and function. So she first conducted separate unimodal models across lots of data available, structural and functional data available in ABCD. And then she selected brain features with non-zero beta weights from those unimodal models and input those into a multimodal model. This is an overview of those findings. Again, I'm not going to go into detail, but you can see here that we found significant associations primarily for cortical thickness and sulcal depth in terms of grey matter. There were associations for a range of white matter tracks and metrics. The strongest associations were for those tracks connecting with frontal regions of the brain. For the grey matter, there's a fairly consistent pattern of higher estradiol levels being related to lower thickness, reduced sulcal depth. And then in terms of resting state connectivity, there were a number of associations primarily between or with connectivity connecting subcortical regions with cortical networks. Just in the interest of time, I'm going to move on to Muskan's more recent work where she's looked at timing and tempo of estradiol exposure in a similar model to the first study I showed you, looking at how these differences, individual differences in puberty, might predict mental health problems via impacts on the brain. So here when I say estradiol timing, I'm referring to the estradiol level that someone has relative to their same age peers. So again, this is just in females. When I say estradiol tempo, I'm just simply referring to changes in estradiol levels over time, so rate of change in estradiol levels over time. So here we were looking at timing and tempo of estradiol, or timing of estradiol exposure at age 10 to 12, tempo, so change in estradiol from age 10 to 12, so that over that couple of years, and then mental health symptoms at age 13. So we kind of tried to tease apart the temporal ordering here for our mediation model. So we looked at global measures of the brain in addition to conducting regional analyses. So in a pattern somewhat similar to the findings I presented earlier, where we were looking at physical development, we found that earlier estradiol timing, so higher estradiol levels relative to same age peers, and also faster estradiol tempo, or greater increases in estradiol over time, were associated with greater reductions in total cortical volume and total surface area over time. I'm showing you the regional effects here. Just wanted to highlight that the complex associations here, there are both positive and negative associations across the brain. However, after correcting for multiple comparisons, most of these regional findings disappeared, and the findings that remained significant were an effect of early estradiol timing on reductions in surface area of temporal regions. So then looking in turn at links with mental health, we found a significant indirect effect involving estradiol tempo. So there were no links between estradiol timing and mental health, but we found an effect whereby faster estradiol tempo, or greater increases in estradiol over time, was linked to higher total mental health problems in the sample longitudinally via reduced total cortical volume. So I'm going to end now just giving you a quick summary of what I've spoken about, and just letting you know about some future directions and other things we're looking at to follow up this work in different samples. So adolescence, we think, is definitely a period of sex differences in brain development, and we have some evidence that this potentially is partially due to pubertal processes. Individual differences in pubertal timing and tempo likely impact brain trajectories with implications for mental health, and this appears to happen in a sex-specific way, or there are sex differences in these associations. With most of the work that I presented, and indeed I think a lot of the work that is out there, showing that links are particularly pronounced for females as compared to males. So I presented some work showing that the social environment might shape brain development via its impact on pubertal development, but the environment may continue to impact the brain in a manner that depends on the stage of pubertal development in a sex-dependent way, or in sex-specific patterns. So an important caveat to the hormone data that's available in ABCD, one saliva sample was provided per participant per assessment wave, and this wasn't controlled, this wasn't kind of at a specific time of day. People provided samples across the day. In all of these analyses, we've kind of controlled for that in models, but it certainly means that the hormone measures in this study are very noisy, and associations are difficult to detect. So something that's particularly important, particularly for estradiol and progesterone and other hormones that cycle, for example, with the menstrual cycle in females, obviously with one sample, we can't look at that at all. So this is what we're looking at in a smaller study in my lab at the moment. We've got multiple saliva samples across the month, and so we're trying to really tease apart, kind of take a closer look at the impact of these hormones on the brain. And with that, I will finish. Thank you to Muskan, Divyangana, Stephanie Hatato, all the people that really drove all of these research questions and analyses, and thank you for your attention.
Thank you very much, Sarah. In the interest of time, we'll only take one question, but you had a couple. So it's when looking at poverty, puberty, and brain development, how is poverty measured, and did you explore psychological distress as well?
What was the last bit?
Psychological distress, did you also explore that?
So the first question, so poverty was a measure of income to needs ratio. So it's the household income adjusted for the total number of people living in the home. So I think we've just looked at, from the brief problem monitor, the self-report version, we've just looked at the kind of total internalizing and externalizing score. That's all we've looked at so far in that work.
Great, thank you very much.